Well, hello, everyone, and welcome to our first of two screencast lectures on the reproductive system. So let's get right into it. First of all, we're going to begin by talking a little bit about functions. Why do we have a reproductive system? Well, first, the reproductive system functions in gamete production, gamete storage, gamete nourishment, and gamete transport. In addition to functioning in these components of gametes, production, storage, nourishment, and transport, the reproductive system is also involved in fertilization. Fertilization is the fusion of male and female gametes to form the first cell of a new individual, and that first cell of a new individual is a zygote. So we all started out as a single cell after fertilization or conception as a zygote. So whenever we take a look at the reproductive system, the primary sex organs that we're going to be talking about, if we're not specifically talking about males or females, we use the general term gonads. Specifically, the gonads are, in males, the testes, and in females, the ovaries. These are the primary sex organs because the gonads, whether male or female, produce the reproductive cells, which we call gametes from our first slide. You may have been wondering what gametes were, and we'll get into that a little bit more as we move along. But the gonads, in addition to producing the gametes, also secrete sex hormones. So the gametes, the male reproductive cells, are referred to as sperm, and the female reproductive cells are the ova or the eggs. As far as the sex hormones are concerned, in males, the sex hormones are called androgens. Androgens are hormones that produce the secondary sex characteristics. When we talk about androgens, the main androgen we talk about is testosterone. There are other androgens, but those androgens are oftentimes referred to as prohormones because they lead to the production of testosterone. So testosterone is the main male androgen. In females, the sex hormones are estrogen and progesterone. But I don't want you to get the false understanding or misunderstanding that males only produce androgens and females only produce estrogen and progesterone. And because what we find is that males do produce estrogen and females do produce testosterone. The difference in secondary sex characteristics, for example, in males and females, however, is that it is the level of estrogen and testosterone. In males, the level of testosterone is higher and estrogen is lower. In females, it's the opposite way around. Estrogen is higher and testosterone is lower. These sex hormones play important roles in the development and the function of the reproductive organs, sexual behaviors and drives, and also the growth and development of many other organs and tissues in the body. So whether you're a male or whether you're a female requires both hormones to be present, but in the different relationships that we just talked about. Next up, the reproductive system not only is composed of the gonads, but there are also accessory reproductive organs that we can talk about. These accessory reproductive organs include ducts, glands, and external genitalia. We'll be taking a look at these from the male reproductive anatomy standpoint first, and from the female reproductive anatomy a little later in part 2 of our screencast. But let's now take a look at the functional anatomy of the male reproductive system. And again, we'll do that in this screencast. And then in our second screencast, we'll take a look at the female reproductive anatomy, functional anatomy. So the male gonads the testes or testicles produce sperm, and they lie within a fleshy sac outside the body wall called the scrotum. Sperm are delivered from the testes to the exterior then through a system of ducts. This system of ducts includes the epididymis, the ductus deferens, the ejaculatory duct, and the urethra. In addition to this duct system, there are also some accessory sex glands. These glands empty their secretions into the ducts during ejaculation. The glands include the seminal vesicles, the prostate gland, and the bulbourethral glands. These glands produce secretions that contribute to the formation of semen. So here's an overview of the male reproductive anatomy that you can refer back to as we go through each one of these different parts of the male reproductive anatomy individually. So let's begin our discussion by taking a look at the scrotum. Again, remember the scrotum is a fleshy sac outside the body wall that contains the testes or the testicles. More specifically, it's a sac of skin and superficial fascia that hangs outside the abdominopelvic wall at the root of the penis, or where the penis attaches to the body wall. It contains the paired testicles, and the testicles are separated from one another by a midline septum. Remember, septa are internal walls, so within the scrotal sac, there is an internal wall, the septum, that separates the testicles one from the other. The external positioning of the scrotum will help to keep the testes at about 3 degrees Celsius lower than core body temperature. This is needed for sperm production. So sperm are best produced and, or I guess I'd just say best produced at a temperature about 3 degrees below body temperature. So when the temperature is a little bit lower, this helps with protein folding and formation, and therefore, when the temperature is a little bit lower, that's what's needed to produce good, viable sperm. If the temperature is too high, it can affect the development of the sperm and cause sperm to be malformed, or if the temperature is too low, it can slow the development of the sperm as well. So we want to keep it right about that temperature, 3 degrees below body temperature of what? 98.6 degrees Fahrenheit, or since we're talking Celsius here, about 37 degrees Celsius. Now the inner scrotal temperature is kept constant then by two sets of muscles that we're going to see here in our diagram. These two sets of muscles are the cremaster muscle first. The cremaster muscle is made up of bands of skeletal muscle tissue, and when the skeletal muscle tissue contracts, it's going to elevate the testis. So here we can see the testicle here, and we can see the cremaster muscle, which is inside the sperm cord, and we can see the organization of the muscles longitudinally this way. So when the muscles contract, they're going to pull the testicle up toward the body wall. In addition to the cremaster muscle, then, there is the dartus muscle. The dartus muscle is smooth muscle, and when it contracts, it wrinkles the scrotal skin. So every male knows, let's say, for example, if he goes swimming, he jumps into a pool of cool water, that would not be ideal for sperm development in the testes. So what's going to happen? The cremaster muscle and the dartus muscles are going to contract. The cremaster muscle is going to pull the testicle up close to the body wall. The dartus muscle is going to pull the scrotal skin up close to the body wall as well to keep the testicles closer to the body where they can be kept warmer. On the other hand, if we're out on a warm day, something like that, and body temperature starts to go up, the testicles will descend, the cremaster muscle will relax and allow the testicles to descend. The dartus muscle will relax and allow the scrotal sac to extend as well, or distend. And the cremaster muscle can relax, but the testicles can't drop further away from the body wall unless the dartus muscle uh, relaxes as well, distending the scrotal sac and allowing the testes to move further away. The testes themselves, whenever we take a look at them, actually have three coverings or tunics. Your PowerPoint may say two, but there are actually three. I apologize for that typo if I didn't get a chance to change it. Now, of these three tunics, we're only going to talk about one that's important here, and that's the tunica albuginea. The tunica albuginea is the capsule that surrounds each testis. So we've been looking at this with the lymph nodes. We've been taking a look at it with the kidney, so on and so forth. We talked about the fact there's always this fibrous covering that covers the organ and separates it from its environment. That's what the tunica albuginea does. It's called the tunica albuginea because it's a layer, and this capsule is white, and that's what albuginea refers to. Now, the tunica albuginea, the white capsule, will have extensions in toward the middle of each of the testicles. These are similar to the extensions that we see in the kidney and that we saw in the lymph nodes. These extensions are called septa, and they extend into the testis, and they divide the testis into regions called lobules. There are about 250 to 300 lobules in each one of the testicles. These septa divide the testicle, as we said, into lobules, and within these lobules we find highly convoluted tubes called seminiferous tubules. So each one of the lobules will contain anywhere from 1 to 4 seminiferous tubules. These are tightly coiled structures, as we can see over here. These are the seminiferous tubules. The white would be the extensions of the capsule, and we can see the various seminiferous tubules there. These tubules are important because this is where the sperm are actually formed in the walls of the tubules, and we'll be taking a look at that as we move along. Once the sperm form here in these tubules, though, then they will be passed out to this kind of beige structure, which is referred to as the ret testis. Once the sperm move into the ret, their next location is going to be the epididymis. So they're going to move from the ret to this kind of comma-shaped structure here associated with the testicle called the epididymis. By the way, speaking of the androgens are going to be manufactured by what are called interstitial cells. These interstitial cells are located here in the areas between the folds of the seminiferous tubules. So that's where testosterone is manufactured. Okay, moving ahead, next up, let's take a look at that epididymis. As I mentioned, it's a comma-shaped, tightly coiled tube, and it's found on the superior part of the testis, and it also runs along the lateral side. So we say it's superior and lateral. The epididymis' function is to act as a place where the sperm can mature until they're ready to be viable and to be able to fertilize an egg. So it functions to mature and store sperm cells. And it can store the sperm cells for at least 20 days. Then, contractions of the muscles in the wall of the epididymis will expel the sperm into the vas deferens. These contractions are akin to peristalsis that we've seen already. So peristaltic contractions essentially will begin to move the sperm once they become viable out of the epididymis and onto the next structure, the vas deferens. So let's take a look at the ductus or vas deferens. This carries the sperm then from the epididymis to the next structure, which is the ejaculatory duct. It is going to pass through the inguinal canal and up over the bladder. So it's going to enter the body wall, and the inguinal canal is located where the lower extremity meets the body wall. It passes through the body wall at that region. It then passes up and over the bladder. Again, the ductus or vas deferens is going to move sperm by the process of wave-like contractions of smooth muscle tissue we call peristalsis. The ductus deferens is located within the spermatic cord. The ductus deferens, blood vessels, the cremaster muscle, which we've already taken a look at, and nerves are all packed inside this connective tissue sheath that we call the spermatic cord. So as we look at our diagram over here, here's the spermatic cord. We can see arteries and veins, and we can see nerves. And we can also see, here's the ductus deferens and the ductus or vas deferens entering the spermatic cord. So that's where it's located. The ductus or vas deferens will end in the ejaculatory duct, which will then unite with the urethra to carry the sperm from the vas deferens onto the urethra during an ejaculation. We should mention here vasectomy. A vasectomy is simply a cutting of the ductus deferens at the level of the testis, and this prevents the transportation of sperm. So a little cut is made. I can't see my cursor. There we go. So a little cut is made right about this level. Stitches are applied and they heal over, and that severs the canal that allows for the transmission of sperm. So the sperm cannot be transmitted during an ejaculation then. Well, what happens to the sperm? If the sperm can't get out of the ductus deferens and the epididymis, then what will happen is after a period of time, they'll be resorbed into the body. So they'll undergo apoptosis and be resorbed. Okay, so here's our diagram of the male reproductive anatomy. We can see the testicle here, or the testis, the epididymis, and we can follow the vas deferens up, and as it enters the body wall, over the pubis, over the bladder, and then comes around here and will form the ejaculatory duct. The ejaculatory duct then will join the urethra as it comes out of this would be the bladder, and join with the ejaculatory duct, and sperm would then be deposited into the urethra and then be released during ejaculation from the urethra of the penis. So the various structures that we just went over there. Turning to the urethra itself, the urethra is going to extend, as we've seen, from the base of the urinary bladder, and it extends all the way to the tip of the penis. The urethra in males carries both urine and sperm, so it's part of the urogenital tract. It's part of the urinary system and part of the reproductive system. Sperm will enter the urethra, as we've seen, from the ejaculatory duct. The urethra is fairly long and extended, so it's divided into regions. The first region is called the prostatic urethra. This is the portion of the urethra that is surrounded by the prostate gland. So here we can see, here is the prostate gland, just where the urethra comes out of the bladder, and the urethra passes through the prostate gland. This is referred to as the prostatic urethra. The next region is referred to as the membranous urethra. This extends from the prostate gland or from the prostatic urethra to the penis. So there's a short membranous right here urethra. And then the final region of the urethra is the spongy or penile urethra. This runs the length of the penis, and that's where it gets its name from. All right, next up, we'll take a look at these accessory glands, the first of which are the seminal vesicles. These are located at the base of the bladder, and they produce a thick yellowish secretion that makes up the majority of the semen. About 60% of the semen is made up of secretions that come from the seminal vesicles. These secretions include fructose, or sugar, vitamin C, and prostaglandins. There are also other substances that can nourish and activate the sperm. The prostate gland, as we've seen in the previous diagram, encircles the upper part of the urethra. It secretes a milky fluid. This milky fluid contains chemicals that will also help in the activation of the sperm. Secretions from the prostate gland will enter the urethra through several small ducts. Next up, we have the bulbourethral gland. This is a pea-sized gland that lies inferior to the prostate gland. It produces a thick, clear mucus. This mucus is important. It cleanses the urethra of any acidic urine, and if the pH is too low, that could damage the sperm as they're being ejaculated or released from the penis. So the bulbourethral gland, in advance of the sperm, or before even an ejaculation takes place, whenever a male becomes sexually excited, the bulbourethral gland will become active and start to produce this thick, clear mucus. Then, during intercourse, the mucus will be released into the penis. It will help to cleanse the penis of acidic urine, and it also will become part of the semen. So the mucus also serves then as a lubricant during sexual intercourse. So it will be released into the vaginal canal prior to an ejaculation. Again, this mucus starts to be produced upon sexual excitement, and even before ejaculation takes place, the mucus will start to be released into the female vagina during intercourse, and that will help with lubrication. The mucus is secreted then directly into the penile urethra. So it's directly secreted into the portion of the urethra that passes through the penis. Again, here's our diagram. We can see here the prostate gland and the many tiny canals that will deliver the prostate secretions into the semen, the seminal vesicle producing its secretions, and notice the ejaculatory duct starts right after the seminal vesicle enters. The tube of the seminal vesicle combines with the vas deferens here. Okay, and then here's the location of the Cowper's gland. So the seminal vesicles, the prostate gland, and Cowper's gland are all going to contribute their secretions to the formation of semen. So what is semen? Well, semen is actually a mixture of sperm and these accessory gland secretions. The advantages of these secretions are fructose provides energy for sperm cells, so it can be used during cellular respiration to provide ATP for the activity of the sperm cells. The semen from the mucus tends to be very alkaline. That alkalinity, as we've seen, helps to neutralize any acid that might be left over from urine production, but it also will help to neutralize the acidic environment of the vagina. The vagina is fairly acidic under normal conditions, and that's important. That helps to uh, keep down the production of pathogens, bacteria, or other germs that might otherwise multiply and divide inside the vagina. So that has to be neutralized, otherwise it could damage the sperm and the semen. Semen also inhibits bacterial multiplication um, within the penis itself. Elements of the semen will also enhance sperm motility. They'll also enhance the ability of the sperm to be motile and to move once ejaculated into and through the vaginal canal. There are prostaglandins in the semen, as we mentioned. These prostaglandins are important. They will help to decrease the viscosity of the mucus. In other words, they'll cause the mucus to begin to thin out in the cervix. So at the base of the uterus in the female, we'll talk about this when we get into the female reproductive anatomy, there is kind of this thick mucus plug that will help to prevent pathogens from entering the uterus from the vaginal canal below. Well, it's pretty thick and a good barrier to entry, and it would also prevent sperm, once ejaculated into the vaginal canal, from being able to enter the uterus and therefore being able to continue its journey to fertilization of an egg. So the prostaglandins in the semen will help to decrease the thickness of this mucus plug and help to make it easier for the sperm to get past the plug and enter into the uterus. Now, if you're wondering about prostaglandins, you could think of them as being very similar to hormones. But instead of being like hormones that are produced maybe from far away sites in the body, prostaglandins are usually produced by cells near the site where they're going to do their work, where they're going to have an effect on cells and other structures. The prostaglandins will also stimulate reverse peristalsis in the uterus. So instead of peristaltic contractions going from the top of the uterus down, what will happen is peristaltic contractions will begin to move from the base of the uterus down at the cervix up toward the fundus of the uterus more superiorly. This will help to draw sperm up through the uterus as well. So this also facilitates the movement of sperm through the female reproductive tract through the ways that we mentioned, by decreasing the thickness of the sperm and stimulating reverse peristalsis. Okay, let's turn our attention to the penis itself. The penis delivers the sperm slash semen into the female reproductive tract. The penis is usually somewhere around 8 inches or so long, and so it's divided into regions. The first region we take a look at is the shaft. The second region is the glans penis, which is the enlarged tip. And on the enlarged tip, we'll see a prepuce or foreskin. This is the folded cuff of skin that surrounds the proximal end of the glans, not the proximal end of the penis, but the proximal end of the glans. The prepuce is what is oftentimes removed or greatly reduced during a circumcision. Internally, we look at the penis and we see that the internal penis, in the internal penis, the urethra has 3 cylindrical bodies of erectile tissue that surround it. This erectile tissue is a spongy network of connective tissue and smooth muscle that is riddled with vascular spaces. That is to say, spaces that have a lot of vascularity, a lot of blood vessels within them. So during an erection, during sexual excitement, an erection occurs. And during this erection, the erectile tissue is going to fill with blood. Now, it doesn't fill with blood just kind of dripping into these vascular spaces. But what's going to happen is the blood vessels in these vascular spaces are going to dilate, and that's going to bring larger amounts of blood into those blood vessels. The blood vessels are going to swell, and they're going to push up against those vascular spaces. That will cause the penis to enlarge and to become rigid so that it makes intercourse easier to occur. The corpus spongiosum is another structure we take a look at, located internally in the penis. This surrounds the urethra, and it expands to form the glands and the bulb of the penis. So here we can see, here surrounding the urethra, we see the spongy tissue that extends all the way through the shaft of the penis, and how it expands down here at the distal end to form the glands and the bulbular portion of the penis. Next, another component of the penis is the crura. This is the proximal end of the penis, which anchors the penis to the pubic arch. So it's not very well shown here, but this would be the crura, the site of attachment to the body wall. Next up, we have the corpora cavernosa as another internal structure. The corpora cavernosa is made up of paired dorsal erectile bodies that are bound to the fibrous tunica albuginea. So here we can see the paired corpora cavernosa on this side and on this side. We can see the vascular spaces there, and it's bound by the tunica albuginea on the outside. So when the blood vessels swell, this tunica albuginea does not distend, and that's what causes the penis to become erect and rigid. And and we can also see, by the way, as I mentioned, that the tunica albuginea now extends from the testes to also form this covering over the internal penis. Okay, so that takes us through the anatomy of the male reproductive system. Now, let's take a look at the functional anatomy of the male reproductive system. And I say functional anatomy because we're not going to get into a lot of detail on specifics on how this occurs, but we are going to take a look at spermatogenesis. That's the sequence of events that results in the production of sperm. So we're going to take a look at the basics of how sperm is produced, but not get into a lot of great detail on this. So as we mentioned, spermatogenesis is going to occur in the seminiferous tubules. It's going to begin at puberty. So when the male reaches puberty, hormone levels will increase and it will activate the process of spermatogenesis. Once spermatogenesis activates at puberty, it will continue throughout the entire life of the male. So sperm production continues throughout the natural life of the male. Now, to understand spermatogenesis, I want to give you a little bit of background information, especially for those of you who may not have had a genetics course or haven't had genetics in quite a while. This will help you to understand some of the basics that we're going to be going over. So one of the things we know is that each cell in our body, we're made up of what, some 60 to 100 trillion cells. Each one of those cells has two sets of chromosomes in it. One set of chromosomes is maternal or coming from the mother, and one set of chromosomes is paternal coming from the father. These are delivered during fertilization by egg and sperm. So most of our body cells are said to be diploid or have the 2N chromosomal number. That 2N chromosomal number is 43 chromosomes. So what we're saying here is that of the 43 chromosomes that we find, and they contain all the genetic information that the cell has, but of those 43 chromosomes, a set of 23 come from your mom and a set of 23 come from your dad. the maternal and paternal chromosomes. So we say we have really two sets. We call that the N chromosomal number. So again, humans have then 23 pairs of what we would call homologous chromosomes. Homologous means similar. You see, the chromosomes that are donated to your first cell that come from your mother contain very similar information as the chromosomes that come from the sperm of your father. What we mean by that is, let's say, for example, the chromosome may contain information about what hair color you're going to have or what eye color you're going to have. Well, you have a chromosome in the set of 23 from your mother that has information about what hair or eye color you're going to have. You also, in the set of 23 from your father, will have a chromosome that contains information about what hair or eye color you're going to have. So they're similar in that respect. But the set you get from mom may say you should have this eye color. The set that you get from your dad might say a different eye or hair color. Then, whenever they're brought together, how they're expressed will determine what eye or hair color you'll have. We're not going to get into the nuts and bolts of that, but I hope you understand what we mean by homologous or similar chromosomes. They contain similar information. Now, whenever we take a look at this, though, we said that we're made up of 60 to 100 trillion cells, and most of those cells contain that full set or 2n number of chromosomes. Gametes, however, the reproductive cells, only have 23 total chromosomes instead of 46. So they have only a half set of chromosomes. This half set of chromosomes is said to be the haploid number, or the N number of chromosomes. Now, this is important, and here's why. If a sperm cell had the 2n number of chromosomes, 46, and an egg cell had the 2n number of chromosomes, 46, then the first cell of a new individual would have what? 92 chromosomes in it. Then, when that individual made reproductive cells, if they had 92, then you'd have sperm cell and egg cell that had 92. You'd see what happened. Chromosome numbers would just keep exponentially growing. So to keep that from happening and too much information being delivered to the first cell of the new individual, gametes have to have their chromosome numbers reduced by half so that they contain, instead of 46 chromosomes, only half that number of chromosomes, which would be 23, or what we call the N number of chromosomes. So how does this happen? How do the chromosome numbers in gametes get reduced by half? The answer is by a process called meiosis. So gamete formation occurs by meiosis, and during meiosis, the chromosome number gets reduced in half from 2n to n. By the way, here is a diagram. This is called a karyotype. This is actually a photograph of the chromosomes from a cell of a human individual. And we can see that the chromosomes are in pairs. So there are 23 pairs. One chromosome in the pair came from your father, and one pair in the chromosome one pair came from your mother. So we have these pairs, and on a karyotype, they're listed in order, generally from largest to smallest, and we can see the 22 pairs here. So whenever we take a look down here, by the way, we can see that one pair here is a little bit different than the rest, where there's one large chromosome. This large chromosome is called the X chromosome, and then there's a small one. This is called a Y chromosome, and this only occurs in males. Males have one large X chromosome and one Y chromosome. Females, these chromosomes right here, would both be X chromosomes. There would be two large X chromosomes. For that reason, these first 22 pairs of chromosomes are called the autosomes, because they're the same in whether you're a male or a female. And this last pair are called the sex chromosomes. Okay, so what's going to happen is in the formation of gametes, the reason why I throw this up is that the chromosome number is going to be reduced by half. So a gamete would only receive one of the two chromosomes in each pair under normal conditions. How does this occur? Well, here's probably the simplest chart that I can find to help you understand how this process occurs. And we'll compare meiosis to another process you may be familiar with, mitosis. Mitosis occurs when one cell duplicates and becomes two cells, and copies of the chromosomes are made, and one copy of each chromosome is delivered to each daughter cell. So let's follow mitosis through first, and then we'll compare that with meiosis. So here we have a cell, human cells, this might be like a human cell, but human cells have 46 chromosomes, right? Well, to show all 46 chromosomes would make this a little bit difficult for us to be able to follow. So let's just take 4 of the 46 chromosomes and follow what happens to them. And here we can see here is one purple chromosome and one smaller purple chromosome. So one long one and one short one. And then we're going to have one green chromosome, long one, and one short chromosome to help us to identify them and follow them, what's happening to them. So we have 4 total chromosomes, 2 purple, 1 long, 1 short, 2 green, 1 long, 1 short. Okay, so during mitosis, what's going to happen is each of these chromosomes are going to be replicated, or they are going to undergo, if you recall your understanding of DNA, they're going to undergo duplication or trans or replication, I should say. Okay, so the DNA is going to replicate itself, and the chromosomes contain DNA, so that's going to replicate the chromosomes. So here we can see this long purple one now is replicated, and the 2 replicated pieces are stuck together here in the center in a piece called the kinetochore. And here we can see the same things happen to the short chromosome, short purple, the long green, and the short green. So these are replicated all along their length, except for the kinetochore. Now, once they replicate this way, this occurs in a phase called prophase, you may be familiar with. Then what's going to happen is the cell will extend these tubules out, which are called spindle fibers. They'll attach to each half of the duplicated chromosome, and they will push the chromosomes into alignment in the center of the cell. This occurs during what we call metaphase. Then, once the chromosomes are pushed into alignment in the center of the cell, these chromosomes will finish their replication and the 2 halves will separate from one another. Now, these spindle fibers which are attached to each half, will contract and pull each half of the chromosome in opposite directions to either side of the cell. This results in, when the daughter cells form, each of the daughter cells having a copy of each of the 4 chromosomes. So it's like during mitosis, what happens is each of these chromosomes, we're going to take the sheet of chromosome, put it on the copy machine, and copy it. So we'll have 2 copies. Here's our 2 copies. Let's follow the purple chromosome, the long one. Here are our 2 copies. Now we're just going to separate the 2 copies, one from the other, and we're going to deliver 1 copy to one side of the cell, 1 copy to our side of the cell. This cell will then elongate and separate into 2 daughter cells, and look at what happens. This daughter cell receives 1 copy of the elongated chromosome, and this daughter cell contains the other copy. This ensures that these 2 cells now will have 1 copy of all the genetic information that's contained on each one of these chromosomes. But what we note here is that this mother cell started out with 1, 2, 3, 4 chromosomes. We would say that's the 2n number. And each of these daughter cells wound up with 1, 2, 3, 4, 1, 2, 3, 4 chromosomes. So they both wind up with the 2n number. This is the way cells reproduce once fertilization takes place in our body. So we start out as a single cell, a zygote. That cell splits, divides to form two cells, and each one of the two cells, because of mitotic divisions, receives the full set of chromosomes. So that this happens over and over again. So by the time that we're, well, who we are today, we're made up of about 60 to 100 trillion cells, and each one of those cells contains the 2n number of chromosomes. Okay, let's take a look at meiosis now on the other hand. We're going to start out with a mother cell. So we're going to start out with a typical body cell. And what's going to happen is that body cell is going to, in meiosis, once again undergo chromosome replication. So we start out with our 1, 2, 3, 4 chromosomes here. And this purple one's going to replicate, and here we can see it. And the long green one, we can see that one. The short green one here, and the short purple one here. But now what's going to happen in meiosis, instead of these chromosomes immediately lining up in the center of the cell, they're going to align next to one another so that these two homologous chromosomes lie next to one another. They contain similar information, and these two homologous chromosomes lie next to one another. This results in two what we call tetrads. These tetrads are formed by a process called synapsis. Synapsis is the process in which these chromosomes will align. So the two long chromosomes will align with one another, and the two short chromosomes will align with one another to form tetrads. We have one tetrad here and one tetrad here. And this occurs during prophase 1 of meiosis. Okay, continuing on, I apologize for that little phone call there. Now what's going to happen is, just like over here in metaphase, in the first part of meiosis, metaphase 1, the tetrads now, instead of the individual chromosomes, are going to be pushed toward the center of the cell, and they're going to align at the center of the cell. Now, spindle fibers will attach to this long green chromosome and this long green chromosome, and to this long purple chromosome and this long purple chromosome. But here's what happens. The spindle fibers now are going to contract and pull the chromosomes in opposite directions. But this time, during metaphase 1, the chromosomes don't completely replicate. So each duplicated chromosome is pulled to the opposite side. So that in our next phase, what's going to happen? Well, you can see here that two green chromosomes were pulled to one side, two purple chromosomes were pulled to the other side. So when the daughter cells formed, what do? They get one duplicated copy of the long purple chromosome in this case, and one duplicated copy of the short purple one. And this daughter cell receives one long duplicated green and one short duplicated green. Okay, now remember, these duplications contain the exact same information. So essentially, this cell gets the information from this chromosome and from this chromosome. And this cell gets the information from this long purple chromosome and this short purple chromosome. Each of these cells now only has, albeit duplicated copies of only two of each of the chromosomes. Now, what's going to happen is these chromosomes, let's take the purple ones here, these purple chromosomes will finish their replication and separate, and they'll be pulled to opposite ends of each of the daughter cells here. So that what will end up happening is, in this second separation of the chromosomes we call meiosis part 2, then each of the daughter cells, look at what it ends up with. one long purple, one short purple, one long purple, one short purple. Each of these has only copies of the purple chromosomes. They have half of the original 4 chromosomes. These 2 daughter cells have copies of only the green chromosomes. They have half of the copies of the original chromosomes. So this is an important concept to understand how the gametes form. And it doesn't matter whether it's sperm cells or whether it's ova, this same meiotic process occurs to reduce the chromosome number in the gametes by 1/2. Okay, now that we've taken a look at that, we can move ahead with spermatogenesis. So during spermatogenesis, cells that make up the walls of the seminiferous tubules will be found in various stages of cell division. These would be mitotic and meiotic cell divisions. These spermatogenic cells will then give rise to sperm in a series of events. This series of events includes, first, the mitotic divisions of spermatogonial cells. These are stem cells, and these stem cells will undergo mitotic divisions, as we just previously talked about on the left side of our previous screen. And those mitotic divisions will result in the formation of what are called spermatocytes. In the second set of events, meiosis will occur in the spermatocytes, and this will form spermatotids from the spermatocytes. So if you're following with mitosis, the spermatocytes will still have the 2n number of chromosomes, but now the spermatocytes will undergo meiosis. Their chromosome number will be reduced by half, so the spermatids that now form will have only the n number of chromosomes. The final set of events in the formation of sperm cells is called spermiogenesis. During spermiogenesis, these spermatids, you could think of them like kids. Kids are immature adults. Spermatids are immature sperm cells. During spermiogenesis, the immature sperm cells, the spermatids, will mature to become sperm cells. Okay, so here's a diagram that we're going to use throughout this process to talk about each one of these sets of events. Okay, so in this diagram, just to kind of set you up here, this might be the wall, the external wall of the seminiferous tubule. And down here, this would be the lumen of the seminiferous tubule. And these are cells attached to the external wall of the seminiferous tubule. These are called spermatogonial cells. These are stem cells. Remember that stem cells are relatively undifferentiated cells that can go on to become specific cells. Here on this side of the diagram, we're showing what's happening with chromosome number. Here on this side of the diagram, we're showing how the cells are moving from the outer portion of the seminiferous tubule toward the lumen of the seminiferous tubule. Okay, so let's start with those spermatogonia. As we said, these are the outermost cells that are in contact with the epithelial basal lamina. So that outer wall is made up of epithelial tissue, and the spermatogonial cells are in contact with the basal lamina, the basement membrane of that epithelial tissue. So spermatogenesis, as we mentioned, is going to begin at puberty, and during that time, spermatogonial cells will begin to undergo mitotic divisions. These mitotic divisions will result in daughter cells. And we are going to label these daughter cells as either type A or type B daughter cells. The type A daughter cells will remain at the basement membrane, and they'll maintain the germline. In other words, type A cells will remain attached to the basal lamina of the outer wall, and they'll continue to undergo divisions to create more A and B type daughter cells. The type B cells, however, will now move away from that external wall, and they'll begin to move toward the lumen. And as they move toward the lumen, they will go on to become primary spermatocytes. So back to our diagram here. So here's the outer wall, and here are the spermatogonial cells attached to the basal lamina of the outer wall. And over here, here is a single spermatogonial cell right here. And here's our single spermatogonial cell. Now this spermatogonial cell will undergo mitosis. Okay, so the chromosome number will remain the same. During this mitotic division, one cell will divide into two cells. One of these two cells, this one right here, will remain attached to the basal lamina. It'll be referred to as a type A daughter cell. The other daughter cell will begin to move toward the lumen. It'll begin to move through what we call the adluminal compartment. This daughter cell that's moving toward the lumen and going to continue to undergo further development into sperm cells, we're going to refer to as the type B daughter cell. So if we look over here on this diagram to help you understand it, here's our spermatogonial cell. It's going to divide to form two cells, two daughter cells. Each of these daughter cells will have the 2n number of chromosomes. This daughter cell, the type A cell, will remain attached to the basement membrane, while this daughter cell will now begin to move through the adluminal compartment toward the lumen. Now I want to be careful here. You see this yellow material that's surrounding the cells? This yellow material is actually a type of cell called a sustenacular cell or a nurse cell. These nurse cells surround the developing sperm cells and help to nourish them and care for them as they develop. But here is a sustenacular cell, and it goes out to here, and look at what happens. Here's the border of this sustenacular cell here, and it comes all the way down here. See that line between them? That's the border between the two cells. So there's one sustenacular cell here and one here. And the reason why I'm pointing this out is I don't want you to confuse this as being a connection between these two cells. This is actually the border of this sustenacular cell and the border of this one. So these cells are developing between the sustenacular cells. Okay, so that's where we're at this point in time. Again, a single spermatogonial cell or stem cell divides to form two daughter cells. Each of these daughter cells contains the 2n number of chromosomes. One remains attached to the outer wall of the seminiferous tubule. It's referred to as the type A daughter cell. The other begins to move through the adluminal compartment toward the lumen. It's referred to as the type B cell. All right, let's continue on. Now, primary spermatocytes will undergo meiosis 1, a first meiotic division, and they will result in the formation of two haploid cells called secondary spermatocytes. The secondary spermatocytes then will undergo a second meiotic division, and their daughter cells will now become, they're only going to have the n number of chromosomes. So the spermatocytes will have the 2n number of chromosomes, but the spermatids will only have the n number of chromosomes. The spermatids will be small and round, and they'll be seen close now to the lumen of the tubule. So let's go back to our diagram. So what's going to happen now is we have this 2n cell, which will now enter into meiosis 1. Once it enters into meiosis 1, it's no longer referred to as the daughter cell type B. it's now referred to as a primary spermatocyte. So here in our other diagram is a primary spermatocyte. That primary spermatocyte will then undergo first meiotic division. This first meiotic division of the primary spermatocyte will result in secondary spermatocytes. That the reason why the number is reduced by half is because each of the two chromosomes, duplicated chromosomes, are going to be pulled apart, not a single chromosome. So that's going to only put one chromosome in one cell and one chromosome in the other cell. So this is going to give us the n number of chromosomes. So once the n number of chromosomes forms, these are no longer called primary spermatocytes, but they're called secondary spermatocytes. The secondary spermatocytes now will have the n number of chromosomes. So here's one primary spermatocyte that undergoes a first meiotic division. Chromosome numbers reduced by half. Each of these now has the n number of chromosomes and is referred to as a secondary spermatocyte. Now, each one of these secondary spermatocytes will then undergo a second meiotic division. This second meiotic division will not reduce the chromosome number any further, and one secondary spermatocyte will now form two early spermatids. So this divides into two cells called early spermatids. This one becomes these two early spermatids. This one becomes these two early spermatids. Again, don't be confused by this line. This is only the border between the two sustentacular cells. Okay, now what's going to happen is these early spermatids will start to undergo maturation to become late spermatids. And that takes us to the next part of the process. So late in spermatogenesis, these spermatids are now haploid, but they're non-motile. They're going to have to become motile. They're going to have to be matured, and they're going to have to become active motile sperm. This happens in a process called spermiogenesis. During spermiogenesis, the spermatids, as they mature, will lose any excess cytoplasm that they have, and they'll form a tail, and that tail will begin to be active and begin to kind of wave back and forth and act as a almost kind of like an outboard motor that'll allow the cells, the sperm cells, to become active and motile. And now they become mature sperm cells. So this entire process takes about anywhere from 64 to 72 days to complete. That is to say, spermatogenesis. Not just spermiogenesis, but the entire process. In other words, from the time that spermatogonial cell divides to form the two daughter cells, and the daughter cells undergo meiotic divisions to form sperm cells, from the time we go from the spermatogonial cell to the sperm cells, that's what takes about 64 to 72 days to complete. By the way, a mature sperm cell actually has three major regions to it. The first region is referred to as the head. The head we can see right here. It contains DNA, and it has a helmet-like structure over its surface. This helmet-like structure contains hydrolytic enzymes. These enzymes will allow the sperm, when it meets up with an egg cell, to penetrate and enter the egg cell. So these enzymes will be released by the acrosome if the sperm cell comes in contact with an egg cell. It'll release the enzymes, and the enzymes will essentially dig through the outer layer of the egg to allow the sperm cell to penetrate the egg and deliver its genetic contents, the DNA or the chromosomes, to the egg. In addition to the head, there is a midpiece. The midpiece contains mitochondria, and these mitochondria spiral around the tail filaments. These mitochondria carry out respiration, which provides the ATP for the tail to begin to become modal. The tail is a typical flagellum as we see in a typical cell, and it is produced by a centriole that we see here. So we have our mid piece here, and then we have our tail extending from there. Okay, so that takes us through the male reproductive anatomy. We're ready to begin the female reproductive anatomy. So we'll stop there in this lecture screencast, and we'll pick up in part 2 with the female reproductive anatomy.